So let's see how can we perform this phylogenetic analysis. So there are multiple steps. So first of all, we will define our problems, or we can have a study group from which we are trying to infer some evolutionary relationships. We do some samplings and we get to our group of interest. We try to get their sequences from the databases, or maybe sometimes we have their sequences which we generate from our labs, and then after that you do those sequence alignments and then you reconstruct your trees. We use the term reconstruction because we believe that these trees or these evolutionary relationships, they exist somewhere out in the evolution, and they exist in nature, but we are trying to reconstruct them. We are trying to make something similar to it. So whenever we are reconstructing those phylogenetic trees, we are trying to infer their actual phylogenetic relationships. So that's why we use the term tree reconstruction or phylogenetic reconstruction. You won't see the word tree construction in the literature. So the main aim of phylogeny reconstruction is to describe evolutionary relationships in terms of common ancestry between different species or organisms or populations. So we can survey the literature and we can narrow down to some group of interest. We might get those informations or the species which we have from our literature search. We can narrow down to some species, or we can narrow down to some genes, and then we can put them into our phylogenetic analysis. So for sampling the group of interest, there must be 4 main sources of data for reconstructing the phylogeny for gene family. We can use published sequences of characterized genes. Remember, those genes which are already been characterized, whose sequences are already there in the which are published, and the sequences are submitted into the sequence databases. We can use gene databases like NCBI. Maybe your data is coming from Express Sequence Tags or EST project databases, so where we have the genes which are expressed and we can have the coding genes in our analysis. We can get some unpublished sequences or genes and we can make up their trees from them. So whenever we get those sequences, we use those sequences and we do some blast searches, and in this way we can come up with some e-values after the blast, and we do some cutoffs. So whenever we are picking up the sequences which are related to our sequences, we will do some e-value cutoffs. So remember, the lower e is the good, so very close to the 0 or sequences are good hits. So we can take that sequence alignment, we put the sequences together and we can view their alignments, or we can do that on some alignment viewers on the computers. We can do that manually, or we can inspect with visually with our eyes, and we can take help with some alignment softwares. There are many softwares. We have seen Clustral W. There is another one, Mega, that is good to do those multiple sequence alignments as well as those phylogenetic analysis. So in this section we see that how can we perform this phylogenetic analysis. So it's the job where you retrieve the sequences of the genes in which you are interested in. You can do some literature survey. There are the databases available. You can get those databases and then you can perform their sequence alignments, and then you can use your phylogenetic tree prediction algorithms or softwares to make their trees.